Hello everyone, myself Samiksha Sontakke, a second prof student at Dr. Ullas Patil Medical College, Jalgaon. Today we are going to study the topic Antisera. So before we begin, let's have a quick glance over the subtopics that we are going to cover under Antisera. Firstly, we will see the definition of Antisera, then the history of its discovery. What are the types of Antisera? Then the preparation of Antisera, mechanism of its action, sources from where we get it, its uses and its side effects. Now let's see what an Antisera is. An Antisera basically is a serum which contains specific antibodies against a specific antigen. Where do we get this Antisera from? We get this Antisera from an immunized host, where this immunized host could be an animal or it could be a human. So we are going to study further in the preparation of Antisera how we get this Antisera from the immunized host. For now, we just have to keep in mind that Antisera basically is a serum which contains antibodies against a specific disease-causing antigen. Now let's see the discovery of Antisera. So Antisera was discovered by the scientist Emil Behring. Emil Behring was working on diphtheria. While he was working on diphtheria, he found that a person who has already got the infection of diphtheria did not get the infection again. Why was this so? This was because of the production of antibodies against the diphtherial bacteria in that person. So, Emil started extracting these antibodies using a guinea pig. And it was in the year 1891 that he saved the life of a young girl suffering from diphtheria for the first time in history. And the significance of Emil's contribution can be marked from the fact that he got Nobel Prize in the year 1901 for his work on diphtheria. Now, let's see the types of Antisera. So, Antisera is of two types, monovalent Antisera and polyvalent Antisera. The word mono means one and poly means two. So, the Antisera which is active against a single specific antigen is known as monovalent Antisera, and the Antisera which is active against two or more antigen is known as polyvalent Antisera. Now, let's see the preparation of Antisera. So, earlier we said that we prepare Antisera using an immunized host. What we do here is we choose a host, inject disease-causing antigen in it, and then in response to that antigen, the host immune system will start synthesizing antibodies against it. Now, we will extract the serum containing antibodies and then process it and purify it. The serum which will be formed will be our required Antisera. Now, say for example, we have to prepare Antisera against diphtheria. Now, the first thing that we have to do is we have to choose an immunized host. So, say here we have chosen rabbit as a host. Now the first thing that we will do is we will inject the diphtherial bacteria into that rabbit. Now this bacteria will act like an antigen for the rabbit's immune system. Now these antigen will go and activate the B cells of the rabbit. The B cells in response will start synthesizing antibodies. Now these antibodies are present in the serum of the rabbit. So we will extract the serum, process and purify it, and thus our required Antisera against diphtheria will be formed. Okay, now let's see how this Antisera is formed. So we know that unless and until an antibody does not bind to the target cell, our immune system won't be able to identify it. And these Antisera contain the antibodies. These antibodies goes and bind to the antigen present on the target cell. Now these target cell is capable of being identified by the host immune system. Now we can see here that the target cell is being identified by the macrophages. Now the macrophage start processing them and present it to the T cell. This T cell start the process of cloning and produces four different kinds of T cell, like the killer T cell, the helper T cell, the suppressor T cell and the memory T cell. The name of these T cells suggests their function, like the killer T cell destroy the antigen, okay, and the helper T cell stimulate the other T and B cell to enhance the immune reaction. Now the suppressor T cells inhibits the T and B cells and it reduces the immune reaction, okay, and the memory T cells remember the antigen. For the next time the same antigen enters our body, our body will show the anamnestic reaction against that antigen. So, where do we get this antisera from? We get this antisera from animals like horse and rabbits or humans. Some of the examples of antisera that we get from horse are tetanus antitoxin, gas gangrene antitoxin, diphtheria antitoxin, anti-rabies serum, anti-snake venom polyvalent. Classification of blood group. Then it is used for the diagnostic purpose in the virology laboratories, for the preparation of antitoxin or antivenom for the treatment of envenomation, to spread passive immunity to many diseases via blood donation. Lastly, we use it in the serum therapy for a specific disease. So these were the uses of antisera. Now let us see some of the side effects of antisera. Local pain at the site of injection. So by this we come to know that we give antisera through injection. So whenever we give antisera, it causes pain at the site of injection. Then erythema and arthritis type of reaction, fever, rash, joint pain and lymphadenopathy. So one more thing that is important to note here is that whenever we are giving antisera, there is always a chance of immediate type of allergic reaction. So to treat that, we should always have adrenaline in hand. So we have covered the theory part of antisera. Now let us recall whatever we have learned by solving some MCQs. So the first MCQ is as follows. The first treatment using antisera was given against option A, pertussis, option B, mycoplasma, option C, diphtheria, option D, rabies. So we have learned in the history of antisera that the first treatment using antisera was given against diphtheria. So option C is the correct answer. Now the second question. At what temperature should the antisera be stored? Option A, -2 to 2 degrees Celsius. Option B, -3 to 8 degrees Celsius. Option C, 0 degrees Celsius. Or option D, 2 degrees Celsius to 8 degrees Celsius. Okay, so antisera is refrigerated at 2 degrees Celsius to 8 degrees Celsius. So the correct answer here is option D, 2 to 8 degrees Celsius. Now the third MCQ. By which route is the antisera given? Option A, oral, option B, injection, option C, sublingual, or option D, nasal. So the correct answer here is option B, injection. Now fourth MCQ. What is the general term used for the collection of antisera against various diseases and toxins for emergency use? Option A, antigen bank, option B, serum reservoir, option C, immunoglobulin stockpile, or option D, antivenom store. So the correct option here is option A, antivenom bank. So that was all about antisera. Thank you for watching my video.